and then that differentiated genome as well. And so this comparison between the level of complexity or potency of a cell and correlating that with which genes are expressed is something that led scientists to start asking, well, if we know that these gene profiles are different in these different cell types, how can we trigger this embryonic stem cell gene expression profile? How could we trigger that experimentally? And that's really what Yamanaka wanted to try. He was also inspired to think that you could do that because Hal Weintraub and colleagues had, in 1987, had a breakthrough where they were able to take fibroblasts, which are connective tissue cells, and they expressed one gene, MyoD, and that one gene was able to convert those cells into muscle cells. So that idea that you could express a single gene which would encode a transcriptional regulator that could change the whole developmental profile of these cells was really monumental. And this gave lots of scientists, Yamanaka included, the idea that if it works for a muscle cell, why can't we, for example here, figure out what would be needed to make something an embryonic stem cell? So his goal was to take a somatic cell, so just a cell from the body, find what these reprogramming factors, and I'm talking now genes, what these genes would be that would make it into an embryonic stem cell-like stem cell. If it's not from an embryo, you can't really call it an embryonic stem cell. So Yamanaka worked with three talented people. There's one paper, which I'll show you in a minute, which he really was what basically won him the Nobel Prize. And that was a two-author paper with Takahashi and Yamanaka.